Hi there, welcome to our IB Biology revision series on section A2.3. This is viruses and it is for higher level students only. So, section A2.3.1, the characteristics of viruses. This is a high level concept. So, if you cast your mind back to 2020, this was when COVID-19 occurred. It happened from an epidemic and then a pandemic. Literally, the whole aviation industry shut down. We were put in, some countries were put in lockdown as well. And when we take some examples of pandemics throughout history, you've got the Black Death caused by the bacteria Yersinia pestis, you've got the Spanish flu caused by the influenza virus, and the Asian flu caused by another influenza virus as well. So, the viruses, they're small in size. Viruses must be smaller than the host cells so that they can enter them. They also lack specific organelles such as the cytoplasm and other structural features. They are a fixed size, so viruses do not grow or do not increase inside. A virus is actually assembled inside a host cell. Nucleic acid is the genetic material, so all viruses have genes made of DNA or RNA, and this is essential as the proteins are synthesized by nucleic acids to a polypeptide translation. So, a capsid made of protein. Before viruses are released from the host cell, their genetic material is packed into a protein coat called a capsid. This is made of repeating subunits. So, self-assembly of the repeating subunit of the capsid gives the virus identical structures, which is different from the living cells. Now, as discussed, there's no cytoplasm, few or no enzymes, so viruses rely on the metabolism of their host. So, the few enzymes which are produced by some viruses are required for the replication of the virus's genetic material for infecting the host cells or for lysis. So, structural diversity in viruses. So, you should have an idea about viruses are highly diverse in their shape and structure. Genetic material can be RNA or DNA. It can either be single or double-stranded. Some viruses are enveloped in the host cell membrane, others are not enveloped. And examples that you need to be aware of are bacteriophage lambda, coronavirus, and human immunodeficiency virus, which is HIV. So, the bacteriophage lambda is a capsid head that protects the double-stranded DNA in the core, the tail fibers that attach the virus to the host cell, and it has a tail sheath that consists of proteins that contract to drive the tail tube through the host cell's outer membrane, and then the DNA is injected through the tail into the host cell. So the coronavirus, this is spherical in shape. It has single-stranded RNA as the genetic material. The envelope is outside the capsid, and it has numerous projections of spike proteins on the envelope, creating a corona. And an example of the host is epithelium cells in the airways and lungs of humans. So HIV, it has an envelope outside the capsid, and the genetic material is two identical single strands of RNA that are protected by the capsid. Within the viral DNA, there is an enzyme called reverse transcriptase, which is encoded and allows the production of DNA using the viral RNA as a model. It is known as a retrovirus because it makes a DNA copy of its RNA code. The envelope spikes of HIV are made of protein and carbohydrate, and an example of a host is T helper cells in the human immune system. So, types of virus and their host cell. So, for example, you've got HIV, host specificity can be white blood cells, poliovirus is spinal nerve cells, hepatitis virus is liver cells, and the novavirus can be the cells of the intestinal system. Thank you very much for listening to this first part of section A2.3. We will continue with section A2.3 in our next recording.